Hi, my name is Jeremy Chai Jian-ning. Together with my partner Cheng Tek Ray, we are going to present about metabolic flux-dependent regulations of microbial physiology. Based on recent studies, change in metabolic fluxes can also trigger phenotypic changes even when environmental conditions are unchanged. This implies that cells can use the rate of metabolic reactions to regulate their physiology. This brings us to the next point, which is cells have mechanisms in place to assess the magnitude of metabolic fluxes. The cells can sense and regulate fluxes mechanistically. Hence, we will further be discussing how flux-based regulations can benefit the cells in some ways. Phenotypes are the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. There are increasing evidence that microbial cells can display phenotype imposed by metabolic fluxes and not directly by extracellular conditions. For example, there was an experiment conducted to test the switching of respiratory to fermentative metabolism in glucose-rich conditions due to flux changes in yeast. It was suggested that the active response of yeast was due to the level of glycolytic fluxes. Metabolic fluxes can do more than just that. As seen in previous examples, metabolic fluxes can dictate metabolic modes. Subsequently, metabolic fluxes can dictate protein expression as well as metabolic fluxes can control growth. Proteomes is the entire complement of proteins that can be expressed. To further elaborate the points made earlier, intracellular fluxes that changes under constant environmental conditions can also reshape proteome expressions. In another experiment conducted, it was found that proteins whose expression correlate positively with glycolytic flux were found to be enriched for glycolytic proteins. Proteins with expression levels negative correlating with glycolytic fluxes, on the other hand, were enriched with proteins involved in the TCA cycle, in the pyruvate, glyoxylate, and dicarboxylate metabolism. This serves to prove that metabolic fluxes can dictate protein expression as mentioned previously. So how does the flux work? Changes in flux induced by environmental changes or stochastic expression of transporters or enzymes could be assessed by changes in the concentration of pathway intermediates. Specific kinetics of the involved enzymes and specific regulation of these enzymes are required. Based on the figure A as shown below, kinetics of the enzymes and their regulation is such that certain intermediates become flux signaling metabolites. The metabolites either correlate or anti-correlate with the metabolic flux. On the other hand, at a constant neutron condition, difference in the metabolic fluxes can be achieved through variations in the neutron transporter levels. This proves the existence of flux signaling metabolite. One such metabolite that was found is the glycolytic intermediate, fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate can either be converted to fructose 6-phosphate through the action of fructose 1,6-bisphosphatase, or it can be acted on by fructose bisphosphatase aldolase to turn into glyceraldehyde 3-phosphate or dihydroxyacetone phosphate.
So we are continue to talk about how the flux works. To transduct the flux information stored in the concentration of the flux signaling metabolite, for example FBP fructose 5-phosphate, into a response, a concentration-dependent interaction between the flux signaling metabolite and other cellular components is required. In fact, it is well documented that metabolites interact with and regulate metabolic enzymes, transcription factors, protein kinase, and cis-regulatory RNA sequence as known as riboswitch that can be found inside the figure. Additionally, some metabolites, such as acetyl-CoA, can have a critical role in the expression of a certain specific genes because they are utilized as a substrate for covalent modifications of histones. Most available information stems from in vivo studies focusing on purified individual proteins or RNA species, in part because direct perturbation of metabolite levels in living cells without off-target effects is still impossible. However, by systematically investigating metabolites that affect transcriptional regulation in vivo, a research showed that indeed flux signaling metabolites such as cyclic AMP, CAMP, FBP, fructose biphosphate, and fructose 1-phosphate, they are interacting with two major transcription factors such as CRP and CRA, and responsible for the majority of the transcriptional regulation observed across 23 diverse growth conditions in E. coli. Also, the interaction of flux signaling metabolites with other macromolecules can exert flux-dependent regulation at different levels. For example, gene expression and enzyme activity regulations. After that, the conclusion is going to talk about why sensing intracellular fluxes. The microbial cells can grow on many different carbon sources, as E. coli can grow on up to 180 different carbon sources. The simultaneous expression of so many sensors probing the extracellular environment will present a significant burden to a cells. Furthermore, signals from different sensors would need to be integrated to ultimately lead to a coherent cellular response. Instead, with intracellular flux sensing, measuring flux at different points in metabolism, for instance, where different inflow nutrients convert through flux signaling metabolites, require fewer sensing mechanisms. Besides that, flux-dependent regulation could also be a strong way to regulate metabolism and cellular processes. First, flux sensing allows cells to determine the actual metabolic rate. Therefore, regulation can be applied on the basis of what is actually happening inside the cells. So that is in terms of metabolic activity, instead of what substrate will be available in the extracellular environment. And maybe because of this, important cellular decisions, for instance the entry into bacterial persistence, are made on the basis of metabolic flux. Second, flux sensing is integrated in global feedback loops as known as flux controls flux, which allow for the correction of random induced alteration in gene expression, which recently was shown to also affect metabolism. After that, this is our reference list, and thank you.